Great. Good afternoon, everyone. Want to get started? Thank you very much for coming. So I'm very proud to introduce our speaker, Manuel. So I actually first met Manuel before I ever actually came to I was down in Medellin, Colombia, where Manuel is from. I was there on sabbatical from FIU, where I was a faculty member at the time. And I gave a talk at the National University that Manuel did some work at. And so I remember after my talk, he came up to me, a younger, nervous Manuel. He came up and he was very excited to introduce himself and talk to me because he had some ideas about using dendrochronology to try and address some of the same questions I've been looking at in the effects of climate change on tropical forests. I've been using tree plots, and he was excited to think about how we can do that using dendrochronology. And I'm not ashamed to say it was love, academic love. For me, at least, I don't know how he felt, but
same way.
I've been thinking about the same questions for a long time. I've been excited about doing dendrochronology, but I didn't know how to get started in it. So to find someone like Manuel who was bright, motivated, and had a lot of experience was just, it was a wonderful opportunity for me. And so we stayed in contact, and Manuel was actually the first student I ever recruited to come and work with me at He wasn't the first to graduate, but since he's been here, he's been a fantastic member of the lab. In many ways, he's been my right-hand man for a long time. As a student, he's faced a lot of challenges. Had to come work, outside of his home country, had to deal with immigration issues, working in a second language. He had didn't have a car. That made some of the work you're going to see a lot more complicated. He had some family drama. Of course, he had to deal with COVID. But despite all that, he persevered through hard work and dedication. And actually, as a result of that, he didn't get to do as much time in Colombia as we had initially thought. It did a lot more work in South Florida than we ever expected. And so actually, all this really cool work that he did in Colombia and tropical forests, none of that made it into his dissertation, and none of that's going to be today. But it's really cool work. So if you get a chance, ask him all about it. Hopefully, we're going to continue working on that. And that's going to eventually see the light of day. But despite all that, and despite this not being kind of the dissertation Manuel expected, it is really cool and important work, and it's definitely something for Manuel to be proud of. And so I am very scared to lose Manuel. So we're actually talking now about ways that we can figure out a way for him to stick around a little bit longer. I don't know if that'll work out, but no matter where he ends up, whether it's here in Colombia, Brazil, anywhere, I have really high hopes and expectations for Manuel. I know he's going to be very successful anywhere he ends up. So with that, I give you Manuel Bernal.
Hello, everybody. Thanks for coming to my talk for my dissertation defense called Evaluating Tree Growth Rates with Dendrochronology Throughout the 20th Century, Effects of Climate and Anthropogenic Disturbances. So even though, first of all, I would like to say that this is my dissertation, it was made with the work of a lot of friends and colleagues that they hate me a lot. Most of you must know that song. So I would like you to keep that in mind and have it as a soundtrack of my talk. And so I will change the name with a lot of help from my friends, because there was a lot of help that I get from them. I can use this. Okay, so my outline, I'll first do an introduction. Thanks, Fred. Then I'm gonna go with chapter 1 and chapter 2 that was made in Miami trees, and chapter 3 finally also in South Florida, but in Big Cypress National Preserve. So trees are exceptional organisms. They occur in different ecosystems. And here on your right-hand side, you can see the biggest tree by volume and height, Sequoia sempervirens. On the bottom, you can see the oldest organism on Earth, Pinus longeva. It is about 5,000 years old. And on the left side, you can see the tallest organism on Earth, Sequoia sempervirens. Also, we have beautiful trees in the cities, such as this bombax tree with beautiful red flowers. Trees are of great importance, given the paper they play in the carbon cycle. and the many ecosystem services they provide. But tree growth is important because through photosynthesis, trees capture CO2 from the atmosphere and store it as biomass for long periods of time, acting as carbon sinks and helping us to regulate climate change effects. So the biosphere is known to be, the biosphere is known to be one of those big carbon sinks in the planet. But there is still a budget imbalance in all these fluxes that we see in the planet. So that imbalance is about the 3% of the total global carbon that we're seeing. But furthermore, trees are being affected by climate change. And one of the ways that trees are affected by climate change is by changing the growth rates through time. So climate change will affect tree growth and therefore the carbon sink the forests provide to the planet. Nevertheless, models suggest a wide range of responses for the mass carbon sink. So in the figure on the left, you can see the land uptake, and you can see the huge variability that the models provide. So the missing carbon sink is still an active research area that we need to go deeper and try to disentangle. Usually, we measure tree growth, measuring the DBH, and we go to the field. One time we measure the DBH, and we come back later, and we remeasure the same tree at the same point, and the difference between those two measurements is how much the tree grew in that period of time. However, this method has a big limitation, a big temporal limitation. So over short time scales, these changes in growth rates could be driven more by local disturbances such as hurricanes or El Niño events than by long-term global change. So dendrochronology offers us a good way to overcome these problems and obtain a better and long-term estimates of tree growth rates in different ecosystems. So tree rings have played a critical role studying temperate ecosystems. And dendrochronology offers us a non-destructive way to assess these long-term changes at an annual resolution, and we can study how the growth rates change in time. So for instance, here we have a period where the trees, with this tree in particular, grew very little, and this other period here on top where it grew a lot. And we can, if we get this data, we can see if those changes in growth are linked to responses to climate, or maybe to fires like this two here in 1980 and 1984, or also to hurricanes or storms. So I'm going to explain a little bit of my method before going into the chapters. So usually we go to the field, and if we are lucky enough, we found fallen trees, we can bring a chainsaw and cut a cross section. But that's not most of the cases. So we use an increment border, such as the one in the middle of the picture. An increment border is a hollow drill that allows us to extract wood cores from the tree without cutting it down and without hurting them a lot. So this is how we do it. Some funny things happen when you're cutting trees. It's not easy and you require some technique. But for instance, and we hit a rotten pocket inside the trunk, and it smells very bad. I had to go and take a shower after that tree. I think it was the last tree of the day. And something related to this is when we hit those rotten pockets, the border can get stuck in the tree and we can lose it. and we will have to abandon it. So some funny things happen when we are on the field. When we are on the field, we not only collect the wood cores, but we also collect some covariables that will help us to do our analysis, such as the DVH, as I showed you before, the geographical coordinates, and we will take some more ecological variables, such as the microhabitat position or the shape of the place where the tree is, or the social position. That means the place in the canopy that the trees are occupying. Next. Okay. Oh, I think I want one more. So once we collect our samples, we go back to the lab, we dry them, we select the better ones. There are sometimes we have rotten samples, as I showed you before, and we have to put those apart. And then the good samples, the one we want to keep, we sand them. And this is how the wood looks sanded when it is scanned at a high resolution. So there are two main ways to do this. One way is directly on the microscope. And wait. Yeah, one way is directly on the microscope, and we will mark the wood under the microscope. But this is the old way to do dendrochronology, but it's a little bit tiring because you need to sit down all day in front of the microscope. So lately, we have, with the development of more technology, we can make high resolution scans of all the samples. And with specialized software, we can measure each of those rings. So this is how the tree rings looks under the microscope. So you can see that these darker cells, those are the lay wood, and this is characterized by cells that have thicker cell walls, smaller lumens, and which give them the appearance of being darker. Whereas the early wood, this one on top is made of big cells with bigger lumens and thinner cell walls, giving it a lighter appearance. So this is what we are looking here in this range, that pattern. So the next step we need to do is a cross-dating. Cross-dating means that we need to find the common pattern between each tree and its population. So you can see on top, each cross is the rings that we mark. We think those were like true rings, but we usually deal with some stuff. So we could have missing rings. Maybe the tree didn't do that ring during that year because it was too stressful that he didn't even have enough energy to make a new layer. But a bigger problem here in the subtropics is false rings. So you can see here we have a few rings that are marked on red, and those are rings that we thought were real rings. But when we start doing the comparison, the cross-dating, we found out those were false rings. So if you look at the panel below, you will see a red line that is the measurements on this sample. And those measurements are following kind of the same pattern that the black line in the background, meaning that they are cross-dating. So both will go up, both will go down, not in the same magnitude, but with the same pattern. And first, we cross-dated some measurements within individuals and then between individuals of the population. So, okay, this is one false ring. And this is another really obvious false ring that at the beginning we didn't even mark it. You see how on the far right, on the far left, the ring comes like very well delimited, but when it hit the blue arrow, it kind of fades away a little bit. So that's one of the ways we have to identify those false rings. And the other ones are the wedging rings, such as that one on the upper left part of the screen, where you can see that two rings come together and form just one. Those are very tricky, and you need to look at them like with tweezers, but hopefully you can pull them apart. Or such as this other ring here that it comes very well defined and then suddenly it fades away too. This is another example of the cross dating. So here we have on top Bursera cimatua, an angiosperm species, and on the bottom panel, Pinus elliottii, a gymnosperm. And in the two boxes surrounded by yellow, you can see the red line, the measurement on each of these samples, following the black line, that is the pattern of the population. So we will consider these two samples cross-dated, and we will bring it to the next step of the analysis. So then we need to standardize the growth. There are different ways to standardize the growth. Basically, all of them fit a function. Could be a mean, a negative exponential, a spline, whatever. And we calculate the ratio between the observed and the predicted values. And lastly, we use that residual to assess the growth or the trajectories to relate to climate change. So, for instance, here on the top, on this figure here on the top left hand, you can see the pink measurements going up and down. The dashed line is the model we adjust, and this little difference here are the residuals that we bring to the next step to assess the trend analysis or to compare with climate data. So, as I told you before, there are a few ways to standardize. One is the conservative trending. This fits usually a negative exponential curve to the measurements, or we can convert those width, because when we measure the rings, we measure the width of the ring. But a better approximation, a better value is to not work with the width, but with the area of the whole ring along, across the whole cross section. Other methods of standardization include the regional curve of standardization, and this is similar to the first one, but here we fit a mean to the whole data together, and we align them by the tangent age. And lastly, we can do the size or age plus isolation, and this is we can do this with the ring widths or with the basal area increments. But basically, what we're doing here is comparing trees that are the same age. So we can compare trees that are 10 years old at different moments, or that are 40 or 90 years old. So now you know a little bit of dendrochronology. So we can start. I can start going deeper in my chapters. So chapter 1 is called Changes in the Climate Suitability and Growth Rates of Trees in Eastern North America. And it was published a couple of years ago. And here on the right hand, you can see the species distribution model we built for Abies balsamea for the 20th century to estimate the probability of occurrence that we will use later on this chapter as a measure of suitability. And I'll go into that. So first, so habitat suitability is reflects the abiotic conditions and maximizes the fitness of a species. But suitability is not easy to measure. So we use species distribution models to calculate the probability of occurrence of these species in Eastern North America. However, we know that there are other factors influencing suitability, as competition, as dispersal limitation, or evolutionary change, or abiotic microhabitat variation. On the right-hand panel, you can see a conceptual model of the fitness suitability hypothesis that basically states that in an environmental gradient, the area of greatest abundance or greatest fitness will be close to the center, and as you go far away from that optimum, the trees or the populations will start to suffer a little bit and have a lower fitness. So we also know that climate conditions have been changing through time. And here on this figure, you can see the y-axis temperature in degrees Celsius, and on the x-axis years. And we can see that in the last 100 years, temperature has been increasing faster than in any other period of time. So for this chapter, we have 3 questions. The first one is, are spatial patterns of tree growth rates in Eastern North America related to differences in climate suitability? So we expect that the tree populations located in areas with high climatic suitability should grow faster than tree populations located in areas with low climatic suitability, as shown here in this figure on the right. So if the climate is better for you, you should grow better. It's kind of the logic we follow with that hypothesis. The second question is, have tree growth rates changed through time in the 20th century? And the third one is, are these changes in growth rates through time due to changes in climate suitability? And for those 2 questions, we have just one hypothesis, and we expect that those changes in the tree growth rates are positively correlated with the species and site-specific changes in relative suitability. So if the suitability was increasing in a site, we will expect that the growth was increasing too, that the growth rates were increasing, or the opposite. So to answer these questions, we download the International Tree Ring Data Bank from the NOAA repositories, and I'm gonna give you an example with this species, Cactus macrocarpa. We did this for 37 species in Eastern North America. So on the left panel and the top arrow or the row, you can see the red dots where that we are here. We see the red dots where the chronology, where those population of trees were. We then convert the ring width to basal area, such as in this figure on the right here. No. ¿Qué pasó? No. ¿Dónde estoy? So we converted ring width to basal area increment, and we correct basal area increment in the y-axis against basal area. To that relationship, we fit the model. That is this purple line. We use a spline here, and then we calculate the residuals by the difference between the predicted and the observed, so we can have a standardized measure of suitability. Each dot in the second panel, each dot here is a tree, and each line here is a population. And then we calculate with a 30-year running window mean growth rates for 1915, 1920, 1940, and so on until we have data for the whole century. Parallel, like at the same time, we download the VN and GB occurrences of those species. So those black dots are occurrences for this special species, Cactus macrocarpa. And we use the Crooklyn time series climate data that spans from 1900 to 2010. So with these two things, we could build these species distribution models that help us to see the probability of occurrence of that species in a given pixel, and then we use that probability of occurrence as a proxy for suitability. Then we extract the values, and because we have the time series over the 20th century, we can have suitability changes also for the whole period of time. Finally, when we have these two data sets together, we join them, and here on the y-axis we have growth, and on the x-axis we have suitability. So that now we have a way to assess changes, how tree growth change based on suitability. Okay, so to assess these relationships between tree growth rates and relative climatic suitability, we use linear mixed models accounting for species, site level effects, and time. So for question one, we fit models that the response to the fixed effect was suitability. In question two, we assess the changes of growth through time. And in question five, we assess suitability and time together and their interaction. We retain different, we build different models to answer question one, to try to hold different responses of these variables. So, for instance, in model one, this helps us to assess the variability across space. This helps us to assess the variability across time, and this across time and space together, including that interaction. And this is with the interaction. So to answer question one, are tree growth rates related to climate suitability? So we found that tree growth is not significantly related to suitability either across space, through time, or both of them together. To answer question two, have tree growth rates in Eastern North America changed through the 19th to the 20th century? We found that tree growth rates are decreasing and trees are growing less through the 20th century. However, we found differential responses on species. So there are some that don't do very well. There are others that don't care that much. And there are others that are happy, such as Fagus grandifolia. To answer question three, how are growth rates changing through time accounting for the suitability? We found that these growth rates are changing through time. As I showed you before, they are decreasing, but that decrease is not related with climatic suitability. But still, the differences among species hold. So Fraxinus nigra still sad, growing too little. There are some others that don't care. And Fagus grandifolia is still doing very well. So to summarize this chapter, our results provide strong evidence that the tree growth rates in Eastern North America are decreasing through time and that are not related to climate suitability, either across space or through time. We observed that the decrease in tree growth rates suggests that we cannot rely on forests to increase their carbon uptake and offset emissions, even in populations with climate change is expected to have positive effects. And the decreasing growth will negatively affect the ecosystem services provided by the North American forests. For instance, we're going to have less provision of timber and less carbon uptake. And this might imply a weakening or saturation of the terrestrial carbon sink. This project was published in Ecography a couple of years ago. So if you want to dig deep on it, you're very welcome. So now we're going to chapter 2, and let's go to South Florida and Miami. Chapter 2 is called Growth Rates of Three Common South Florida Tree Species Are Being Affected by Climate Change and Urbanization. And I want to thank especially to undergraduate students, Lauren Combs, Laura Garriga, and Andrea Rivera, who helped me measuring the tree rings for this project. So Miami is located in the subtropics, and this is a strategic place because it's an ecotone between the tropics and the temperate zone. So here we have some temperate species that are growing at the southern edge of the geographic distribution, such as the Pinus elliottii, splash pine, Tercumbia virginiana, live oak, or Arces rhodum, the red maple. But we also have tropical species at the north of the geographical distribution, such as the Sutenia mahogany, the West Indies mahogany, Bursera cimarua, known as gumbo or Terminalia catapa, just to name a few. So we select 3 species occurring in South Florida, one from a temperate origin, growing at the southern extent of their geographic distribution, like Pinus elliottii, and 2 from a tropical origin, growing at the northern extent of their geographical distribution, such as Gumbo limbo, Bursera cimarua, and Sutenia mahogany. So with the permission of the Miami-Dade County Parks Office and Florida State Parks, we visited the 16 parks on South Florida. We didn't find all species in all parks. However, we collected between 31 and 35 samples for those species, which adds up to 100 trees. We download the climate from the Florida Climate Division 6 from the NOAA repositories, and we construct this demograph that you can read on the left y-axis. You can see temperature, and you read it on the red line, and on the right y-axis is precipitation, and you can read it in the blue bars. And the x-axis shows each month. So basically, in South Florida, we have hot, wet summers and dry, cold winters. So we divided these parks into main habitats: natural preserves and urban parks. And natural preserves don't have active management. Usually they are closed canopy, no pruning, no fertilizing, or any other type of management, whereas urban parks are open areas with a lot of land subjected to a lot of gardening practices. And this is how the pine rocklands, the natural park of a pine rockland looks like. And this is another example of A.B. Barnes Park. And if you still, I forgot to mention this, but this was my poet project. And one nice thing, or one thing that happened to this project, is that I bike to all these locations from campus to all these parks, and this is my evidence. So for this question, for this project, we have 3 questions as well. What are the climatic drivers of tree growth in these 3 South Florida tree species? So we think that trees at the northern extent of their species geographic range are more likely to be limited by cold temperatures, whereas trees in the southern portions of the range will be commonly limited by abiotic interactions, low water availability, or high temperatures. Our second question is, are growth rates of these species stable or changing over time? And we think that growth rates will be decreasing over time in Pinus elliottii, but increasing in Sutenia mahogany or Bursera cimarua because of the geographic condition of their location. So Pinus elliottii is in the hottest part of the range, so a little bit more temperature increase will bring him out of his comfort zone, and he will start to struggle. Whereas for Swietenia mahogany or Bursera cimarua, that are tropical species and that are growing in the coldest part of their distribution, that little increase of temperature could mean a little boost for the growth rate. The last question for this project is, are these tree growth rates affected by urbanization? And such as the previous hypothesis, we expect that this relationship will be very related to temperature, and we think that Pinus elliottii will grow slower in urban parks, whereas Bursera cimarua and Swietenia mahogany will grow faster in urban parks than in natural parks. So these are the chronologies we built. On the top is Bursera cimarua. The oldest Bursera gumbolimbo that we found was 125 years old, and the intercorrelation is 0.46. In the middle is Pinus elliottii. The oldest pine we find here was 131 years old, and the correlation was 0.5. And lastly, the Swietenia mahogany was the oldest species that we could find for this project, 167 years old, and the correlation in all those samples was 0.46. So to answer question one, what are the climatic drivers of tree growth in these South Florida tree species? We did a correlation analysis with the current and the previous year. We analyzed each month against our chronology to see which of these months was influencing the most the growth. So we found that for our two angiosperms, they had a positive correlation with the fall precipitation, meaning more precipitation in the fall will mean that these trees will grow better. We didn't find any relationship or correlation with the temperature for this species. And when we look at the previous year, we found a very strong response of the three species. Basically, negative correlations between the precipitation of the previous first quarter of the year and tree growth. So they don't have the same response, but we see that Bursera cimarua had a negative correlation with March precipitation, as well as the Pinus elliottii. And Pinus elliottii also had a negative correlation in August. And the summatory of all the precipitation in that first quarter of the year. Also, the negative correlation of February with Swietenia mahogany in that same respect. And two correlations with temperature, one positive in Pinus elliottii and one negative for Swietenia mahogany. To answer question two, are the growth rates of this species stable or changing over time? We did a class isolation analysis. So this means I'm going to explain it the first figure here on that says age class 10. So each of those points in each panel is a tree when that tree was 10 years old. And on the y-axis, you can see growth rate in millimeters. So this is width. And on the x-axis, there is time. So we see that over time, these young trees are growing faster. So 10-year-old trees in 2020 are growing faster than 10-year-old trees in 1900. And we found that these species ages cause specific ontogenetic responses of growth rates to changing climates. And as tree gets older, we reach this point when they are 90 years old. And here the pine and the Pinus elliottii start having significant negative relationships, meaning that these old trees are starting to suffer with the changes that are happening in climate here at the end of the study period. So our third question is, are tree growth rates affected by urbanization? And we found that annual growth rates respond differently between the species based on their geographic ranges and their habitat. So species with a tropical origin show positive responses to that temperature increase in the urban areas, whereas the pine, no, whereas the pine show a negative responses in the urban park, showing that the temperature difference in urban parks versus natural preserve is affecting negatively the tree growth rates in these species. When we analyzed, or when we went deeper to see if the natural preserves and the urban parks indeed have differences in temperatures, we found a significant difference in the temperature, in the mean temperature of those parks, meaning that a lot of the response back here is driven by the changes in temperature, although we cannot put apart those effects completely. So to summarize, the foundational species of the endangered pine rocklands and hardwood hammocks might be vulnerable to changes in precipitation and temperature, as well as other environmental effects associated with urbanization. We found evidence that the species growth rate can be resilient to changing climate conditions, which indicates that there might be other processes helping the plants control how they grow and how to regulate stressful conditions. And we expect that information about the climate response, growth rates, and age structure of these species will benefit protection, conservation, and restoration efforts in South Florida. And this research was submitted to the Journal of Forestry Research, and we're waiting for their response. So lastly, chapter 3, still in South Florida, but west from Miami. This chapter is called a dendrochronological assessment of growth rates in three dominant tree species of the Big Cypress National Preserve in Florida, USA. And I want to acknowledge and thank the UM undergraduate student, Cici, that is here with us today, and Ari O'Reilly, that helped me measuring these tree rings. So, Taxodium ascendens, the dwarf cypress, Taxodium disticum, the bald cypress, and one species of pine, the slash pine, dominate the landscape and in Big Cypress. However, Big Cypress constitutes the largest population of dwarf cypress, and it covers up to 25% of the preserve. So, however, this preserve is subject to a lot of pressures, including changes in climate, hydrological patterns, hurricanes, and storms, and anthropogenic pressures such as the constructions of the highway, the bridges, the canals, and also the establishment of Big Cypress as a preserve in 1974. So geologically, Big Cypress is on top of a limestone, and the bedrock is formed by a few geological formations. Big Cypress is almost flat, as you can see it in the photo, and there is a gentle slope from northeast to southwest, and the maximum elevation there is just 7 meters above sea level. Super high, hard to hike. And the combination of these geological materials and water flows have created a mosaic of solution holes in the landscape. So if you go close to the solution holes, you can see the water here from the ground, and around those solution holes, the Taxodium disticum are established. And as you go farther away from the solution holes, you start finding taxodium ascendens or dwarf cypress. Whereas the pines are restricted to these 3 islands that are just a few centimeters higher than the rest, but they are never flooded. So the soils in big cypress are usually not consolidated substrate, so that means they are very, very poor. And these could be formed by rocks, carbonate, marl, sands, or organic soils or pits near the solution holes. And there are 5 types of vegetation, including hammocks, cypress domes, cypress strands, prairies, and pines. And this is how the cypress dome looks when you do a cross-sectional cut. So here in the middle, you have the solution hole, usually full of water all year round, and full of alligators around it, the big cypresses that are taxodium disticum. And as you walk far away from the hole, from the solution hole, you find the small cypresses that are taxodium ascendens. So this is the location of our study site. On the right hand, you see the Florida Peninsula, and in the red box is big cypress. The yellow lines are the trails inside the preserve. And to do this sampling, we took advantage of the Florida Trail, and on I-75 Alligator Alley, we got in on that direction, on southern from I-75 up to 5.5 kilometers to sample these trees. We download the climatic data from the PRISM group, and we build this climograph here. And again, the pattern is kind of the same that in Miami. We have hot, wet summers and dry, cold winters. And on the bottom, you see the climograph, but for the water levels running through Big Cypress. And we see that the peak of water depth is in is in September, 3 months after the peak in precipitation that is in June. So these are the species we selected. The Pinus elliottii growing in the 3 islands, the taxodium ascendens going away from the solution holes, and taxodium disticum, bigger trees next to the pond, all around together. For this chapter, we have 3 questions as well. The first one is, how are the growth rates of Pinus elliottii, taxodium ascendens, and taxodium disticum related to environmental variables such as climate and hydrology? We think that tree growth rates will be negatively related to temperature and vapor pressure deficit. However, precipitation and water levels will be positively related to tree growth because more water can increase the length of the growing season. Question 2 is, how are these species growth rates changing through time? And we think because of the geographical location of these 3 species, these growth rates will be decreasing through time since they are in the hotter part of the range. And lastly, how do growth rates of trees change across the Big Cypress landscape? And we think that the taxodium species tree growth rates will be higher when the trees are closer to when the trees established closer to the solution hole, whereas the Pinus elliottii growth rates will be higher when they are farther away from the edges and like close to the center of the islands, so farther away from the water levels. So we went to the field in 2 years ago. We did 3 field trips. We camped in Ivy Camp, and we collected 80 individuals, 27 Pinus elliottii in 10 tree islands, and 53 taxodium trees in 9 cypress domes. So here are the chronologies we deal for these 3 species. In general, the trees in Big Cypress are way older than the trees in Miami. So the oldest Pinus elliottii was 162 years old, and the correlation in all the time series was 0.43. The Taxodium ascendens, the oldest tree we found was 289 years old. I think that's the oldest tree in all my collection and the oldest tree I measured in all my land. Even though if you tell our endocrinologists, it's not too much. They want more than 500. Someday I found one of those. The correlation was good, was 0.42. And lastly, Taxodium disticum. The oldest Taxodium disticum was 219 years old, and the intercorrelation was 0.4. One important thing we noticed on these 3 chronologies is a decrease of growth in the last 10 years, like repeatedly in the 3 species. So our results, to answer our first question, how are the growth rates of these species related to environmental variables such as climate and hydrology? We found that tree growth is driven by the previous year water level, and the 3 species respond very strongly to them, although in different times of the year. So Pinus elliottii is more correlated with May and June water flow, whereas Taxodium ascendens likes to grow better during June. Although if we take into account the mean depth between June and September, the mean depth of the summer, it is also its tree growth rate also correlated with that and shows that the more water it flows during this period of time over Big Cypress, the better they grow. And the panel is even stronger in Taxodium disticum, where we can see that it correlates positively and significantly from May to October. To answer question 2, how are species growth rates changing through time? We did the same analysis that I showed you in the last chapter, but for this analysis, we converted the tree ring width to basal area increments. So these are square millimeters. And we did the same analysis. So each dot here is a tree when it was 10 years old, 30 or 50, etc. And we found that the tree growth rates change through time. Taxodium disticum and Taxodium ascendens have positive significant slopes when they are young, which leads me to think that these are young populations that still have increasing growth rates. And you will say, oh, but you show us a 280-year-old tree. How you say that's a young population? Well, these Taxodium species can reach up to 2,000 years. So I still think that these are young trees, and we are, well, and the trees we have there is like a succession of previous disturbances. And we found that despite those positive growth rates when trees are young, when trees reach all their ages, they start growing slower. And lastly, how do growth rates of trees change across the BCI landscape? So to do this analysis, we average the tree growth rates. So each point is a tree, and we measure the distance to the edge of each tree, to the edge of the tree island in which it was growing. And we found that indeed the pines like to, oh no, the pines grows better the farther in they are in the island. We did the same for Taxodium species. So this is Taxodium ascendens, and what we found is that taxodium doesn't care about being close to a solution hole or to the center of the dome. That slope is pretty flat, but we found a marginally significant relationship of taxodium disticum, of the pre-growth rate of taxodium disticum, and the distance to the center of the dome, highlighting like the wet preferences of this species. So to summarize, growth is driven by previous year water levels. There is little influence of atmospheric variables due to other disturbances. Trees in the region of this region of Big Cypress have been exposed to a suite of disturbances during the last 300 years. For instance, canal and road construction, hurricanes, logging. And we found that tree growth rates change based on the distance to the domes for taxodium species and of the edges for and edges of tree islands, Pinus elliottii. Finally, the tree growth rates of Pinus elliottii are decreasing through time, suggesting that the southernmost edges of its distribution is starting to get too hot. So to conclude, I want to say that my dissertation provides strong evidence that tree growth rates in Eastern North America are decreasing through time and are not related to climate suitability. We found that the foundational species of the endangered pine rocklands and hardwood hammocks might be vulnerable to changes in precipitation and temperature, as well as other environmental effects associated with urbanization. And we found that tree growth in Big Cypress is driven by water levels and that the relative position of the trees within the landscape. Finally, overall, my dissertation showed that trees in Eastern North America and South Florida are growing less through time unless they are young populations, such as the one growing in cities, or old populations that are being reactive to other disturbances. Therefore, trees are losing their potential as carbon sinks. And this research opens a way to think about how forest management can help us increase CO2 sequestration and storage for long periods of time in both natural and urban ecosystems. So I'm gonna switch to Spanish. Quiero agradecer a mi mamá y a mi papá, quienes siempre me han apoyado en todos mis proyectos, a pesar de lo locos que parezcan. Sin su ayuda y su apoyo incondicional no hubiera llegado a donde estoy hoy. Quiero agradecerle a mi hermano por enseñarme a ponerle música a la vida. And soon the trees will sing. I want to talk to my son Jerónimo. I wanna thank my son Jerónimo. You are my inspiration every day. I still cannot believe that you are attending my doctoral dissertation defense and that you understood most of the things I said. that makes me more proud that being here. Thanks for accepting the challenge of a school interchange, and for your company. It's been amazing to live with you again. Hero is an amazing goalkeeper, pianist, and he also likes to come to the forest with me from time to time. I want to thank to my friends in the biology department. Not everybody is in these pictures, but we did fun things and support important causes. I highly value the beers, talks, and time spent at the veranda afternoons. I want to thank Alisa, because you helped me through the hardest part of my time in Miami. And getting to know you and making our friendship grow is one of the best things that I'll keep from this stage of my life. You have been incredibly supportive, personally. I want to thank Riley and the Sierra Nevada Santa Marta crew for accepting the challenges of working in Colombia and be always down to do cool trips that turn into cool science. I hope that this is the first of many collaborations together, and it's a pleasure to work with you. I want to thank to my friends in Colombia, El Salpicombo, for the for their friendship along the years, and to Bob Marley. I'm gonna read the next two. I want to thank Daniel Zuleta for his time and dedication in teaching me how to code. For your guidance and help during the period was fundamental of the rest of my PhD process. I want to thank to my collaborators, especially Courtney Angelo from Cypress National Preserve. Your help preparing the grant application was essential to make it a grant-winning project. Winning this grant offered me the opportunity of working an academic year as my own research assistant and gave me the possibility to work exclusively in my research, stay longer in Colombia, and share more time with my son during that specific year. I want to thank Dr. James Spears for inviting me to come to the course Turing Science and its applications in Zambia. This course boosted my chronological skills and gave me the tools to develop two of my dissertation chapters. I'm very thankful to Jim for inviting me as a visiting scholar to his lab in Indiana State University, and for teaching me in detail the basis of dendrochronology. I also want to thank Dr. Stockton Maxwell and Dr. Nicole Sampieri for sharing their expertise during the course in Zambia and for replying to my emails promptly when I reach out asking for help. And I want to thank to my advisor, Ken. Thanks for believing in me more than I believe in myself. Giving me the opportunity of doing a PhD when I was 36 and being my academic advisor. Thanks for your support and for allowing me to put life over work. Understanding that family and friends comes before work, and even knowing that projects will be delayed. Since the first day that I came to Miami, you made me feel part of your family, letting me stay at your place while I found one. You are an admirable person and scientist, and I don't have words nor time to thank you for all the things you have done for me and Jeromy. I want to thank to my lab mates, former and current members of the Jungle Biology Lab, and all the collaborators and my committee members and research sponsors. Thanks a lot, and I'm happy to take any questions. Great.
Your conclusions for chapter 1 cover a pretty wide range of growth rates.
I have one.
You know, pretty get a pretty widespread growth rates. So species growth rates for very, very negative.
Yes.
I don't know if you've done this analysis like for real yet, but just looking at that, this is species. Do you found that there's any like trait that seems to explain the spread?
And we did one step further in that research.
Yeah.
And it was separating gymnosperms from angiosperms. It was the only thing that we have enough power to put apart. We could have done it with the genes or family level, but it was not enough power. And we found that gymnosperms are doing worse than angiosperms. So because their wood anatomy is a little bit different, less specialized, they tend to, they are suffering more than angiosperms. But yeah, it's a wide range of responses, and some do better than others. But what I would like to emphasize is that in average, the trend is decreasing. Thank you. You're welcome.
Yeah, so in chapter 2, some of the parts, like Doug Barnes Park, would have the mahogany in the natural area and in the more manicured area.
Yes.
Did you get enough samples to be able to compare, like within a single park, natural versus urban, or?
No, within a single park. Indeed, to sample mahogany was really hard to find a natural stand of mahoganies, and we went down to Key Largo, because all the mahoganies we found here in Miami are like in parks surrounded by lawns, or in the middle of the streets, or like in the urban park setting. So that would have been very nice to do, like try to put those effects apart, but no, we couldn't.
That was a great.
Sorry.
Covered all the trees I planted in my yard. I wanted to ask about the, I thought that your findings for the edge effect on the slash line were great. It seems to me that implies that any, we have a lot of squares to set aside in slash line in the city. It seems like thinning, whether it's natural or it's controlled fires, that also have a really detrimental effect with opening up those plots. They would appear that any effect in the plot itself. And that's an observation. But the second part, this is primarily heat today.
We couldn't put those things apart. I would argue that it's a combination of urbanization effects, and it is a very important one there. But in saying that, it could also be that there are microorganisms in the soil, or different soil conditions, or different effects that we couldn't control all in this project. But yeah, I think that the effects on the pine are serious, and it's something to worry about, even more when Miami Dade County start a project to replant a lot of native vegetation around the city. So I don't know if you have seen that there is a lot of new baby slash pines all over campus and in a lot of neighborhoods. So if we want to ameliorate that negative effect in the pine, we should start thinking on how to develop better open space and better parks, not only for us, but also for the city, so they can do it better and don't like trying to give us something.
Would you recommend replanting those edges that are dying back? I've seen just a lot of edges that have died back.
But if you plan, if you fix the edge, you won't do anything. The edge is on the edge effect all the time.
It's always a bit.
I will try to grab like the whole park and start switching lawn and cement, or I don't know, like for kind of an understory type of vegetation, with shrubs, some grass, and more layers, so we can mitigate.
I had one other question. I have a lot, but one other simple one. Is there an easy way to just look at a cypress and figure out which one it is, or do you have to look at its habitat?
No, that was very hard for me, and I'm not a taxonomist, so for that project I invited Ryan. Right, a lot of help from my friends. And he is a good taxonomist. So basically what the literature says about cypresses, some people say there is the same species and different varieties, and some other people say there are still different species. But one of the characters to put them apart is the size. So a dwarf tree is not more than 3 meters tall, whereas a ball cypress is to be 20 meters tall. And the other thing is in the leaves. So taxodium ascendens is called ascendens because the leaves are going like upwards. And taxodium disticum is called disticum because the leaves are like on a disticum position along the little branch. So it's not easy, but there are tools to tell them apart.
Can they grow together, or do they grow heads?
Well, when you are in the domes, so when you are in the domes, you see those little bumps, those are formed by the taxodium disticum. The other taxodium ascendens will be here on the other edges of the domes or in the open areas. So there is also like that location. You can also use the location, put them apart. But at some point, there will be a mix of them, like on the edge of the head on. And that's when Riley was.
I think I'm hopeless. I put my encrypted seeds from outside for that part of the craft though. It didn't come from like the part. Great talk.
Riley,
Yeah, I had just a general question about methodology, I guess. In the very beginning, you said that one of the covariates that you collect is like position and canopy. And this question applies to different technologies. But does that have a kind of explanatory power in the models, since that position and canopy changed through time? Like 5 years ago, it was in shade, and now it's in the sun.
Well, and yeah, that's a big problem for the endocrinology, because yeah, we can go to a field and say, okay, the tree is in the canopy right now. And we have 1 point observation, whereas with endocrinology, we have a long history. So it won't be super powerful and super explanatory having just 1 observation. So the ideal thing would be go several times and try to see how the tree is doing its way up to the canopy. But I have read some researches that have found differences between trees growing in the understory and trees growing in the canopy or on the upper layer. And usually trees in the understory have more have a smaller growth rate. So if they don't die, they tend to be older than big trees.
And is it really easy to see like when, like a point in time when a tree maybe fell down and then now this tree is growing a lot faster?
Yes, there is one kind of analysis that you can use to identify those kind of releases.
Yeah.
So the tree will be without competition, it will release like a shot of growth. It's okay, it's my turn. And you will see something like the rings I showed at the beginning that were very wide, something like that. But this,
I was wondering about that one, so regular sequence the time. What
that's a very good question. It's not suitability. That's what we have to say from that research. More than that, I would say they are struggling with climate change. But with that paper, I don't have enough evidence to say. And the other option is that they are becoming more water user. So they are growing less but they can't handle any over they get better. That to be like for explanation for that result.
But if you have the forest getting older as well, and I'm seeing one of these forests regenerate.
Can you repeat that?
And the forest is getting older in general, the trees are all getting older.
Yes, but it could be that if we don't standardize the data. But when we standardize, we are taking into account the age and the size of the tree. So it's not good.
So thank you, Manuel. Next questions online.
Yes,
Good job.
thank you. Congrats, amazing job.
We'll go to my lab. for questions. If you got a break or anything.
Hey, I was just gonna bring up. Okay, no, I was talking about you.
Yeah. We'll go over to my lab.
Yeah. Just breaking one is-
Yeah, for the group. Thank you. Come on. I thought I